again, one of the things we know about the digestive system is that, and we had talked about this morning, that the digestive system itself can be an integrating center, but so can the central nervous system. So when we think about the way that we regulate the movement of food through the gut and stuff like that, we have to think about that. And we also divide the digestive process into phases. Some of them are pretty self-explanatory, like the gastric phase is what happens in the stomach. The intestinal phase is what happens in the intestine. But the first phase of the digestive process that we have is the cephalic phase. What does that word cephalic mean? In your head, right? Okay. So, the obvious answer to what happens in the cephalic phase is what happens when we put food in our mouth, right? But there's more to it than that, because that's not the only thing in your head. We've also got a brain up there as well, right? And we said this morning that the brain is involved in the digestive process. So the cephalic phase, of course, involves chewing our food, which we'll talk about in just a couple of minutes. But you actually are starting the digestive process before you even take your first bite of food, right? And I think that this is most apparent when we're hungry. But when you are hungry, it's getting close to lunchtime or something like that, all you have to do is smell food or think about food or talk about food or see it. And your stomach starts to grumble, you start to salivate, do all of these things, right? So the cephalic phase of digestion begins before you actually take your first bite of food. When those things happen, by the way, when you start to salivate because you're thinking about food or something like that, those are called feed-forward reflexes. And what your brain is doing is it's sending messages to your digestive tract to get it ready for a meal, right? Starting to secrete saliva. Although you aren't aware of it, you're starting to secrete stomach acid and stuff like that at that point as well, right? So those feed-forward reflexes are really important for us to kind of prep ourselves for digestion. Kind of an extreme example of that is snakes. Okay, there's certain species of snakes that don't feed regularly. There are these snakes that can go literally days, weeks, months, or in some cases a year or more between meals. And what they do is they will actually during those long periods of time between meals, what they'll do is they'll actually let their digestive tract almost completely atrophy. It almost completely goes away. Your digestive tract is a fairly large set of organs in your body. And to maintain those tissues and those organs costs a lot of energy, right? Kind of like having to spend money to maintain your car, right? And if you're not using it for a long period of time, it makes sense that you wouldn't want to spend that energy to maintain it. So certain species of pythons will actually let their gut, rather than spend the energy to maintain it, they'll actually let their digestive tract completely waste away between meals. And you just save a lot of energy in terms of maintenance for that. But the problem is that you need to get it ready for a meal. So you start to salivate, your stomach starts to grumble. They have to rebuild their entire digestive tract in order to be able to prepare themselves for a meal. So a couple of days before they eat, their metabolic rate skyrockets, and they completely rebuild their gut. It takes so much energy to do this. They draw on fat stores that they have inside of them. Snakes have a really large body of fat inside of them. And they'll start drawing on those fat stores. They actually pull so much fat out of that fat body while they're rebuilding their gut that their blood plasma becomes milky white. So if you were to take a blood sample from a snake in the few days before it eats, its blood would be a milky white color because it has so much fat in its blood. It's really, really remarkable. If you ever had a patient, a human, or with milky white blood plasma from all of the fat in it, you definitely would be sending them straight to the emergency room, I would imagine, because that would be a really bad situation. But snakes do this on a regular, at least some species of snakes do this on a regular basis. So it's really, really cool. And then they rebuild their gut, and then they are ready to eat a meal. You and I don't do that. We maintain it continuously, but we do have these feed-forward reflexes that allow us to prep ourselves for a meal by secreting saliva, by secreting stomach acid, and everything else. And of course, the vagus nerve is heavily involved with those feed-forward reflexes because that's what goes from the brain to the stomach, right? So the vagus nerve is heavily involved. Let's take a look at what happens in the mouth during the cephalic phase of digestion. The first thing that's worth noting is that we have two types of digestion. This morning, you guys told me that in order to break down fats or proteins and break these chemical bonds, we would need enzymes. That's a type of digestion that we call chemical digestion. Okay? It's called chemical digestion, and it's named chemical digestion because you are breaking chemical bonds when you break down proteins into amino acids. When you break down starch into the glucose molecules that make it up, you're breaking chemical bonds. Another kind of type of digestion that we have is called mechanical digestion. So we have chemical digestion and we have mechanical digestion. Mechanical digestion is simply just breaking down our food, right? And the most common way, the most obvious way that we do this is by chewing it, right? We're breaking it into smaller pieces so that the enzymes can do their job. So in mechanical digestion, we're actually making our food into smaller pieces, but we're not breaking any chemical bonds in the process. Like I said, chewing is a really, really good example. So in our mouth, we do two of those things. We secrete saliva, and that saliva is going to aid us in digestion. But we also are chewing our food, which is a form of mechanical digestion. Okay? So what does our saliva do? All right, let's go ahead and take a look at our saliva first, then we'll take a look at some of our teeth. Our saliva does a number of things. One of the things that it does is it just simply moistens our food. It softens it. It makes it easier for us to swallow. Right? Kind of binds that bite of food into a bolus that allows that we swallow all at once. Right? By dissolving our food in a fluid medium, it makes it easier for certain chemicals to bind to taste receptors. So our sense of taste is better when we have saliva. If you've ever had to eat with a super dry mouth or something like that, usually when we're sick we experience those sorts of things. But if you've ever had to eat with a super dry mouth, you notice you don't taste a whole lot, right? You've got to have that food moistened to be able to do that. We also have a substance called lysozyme in our saliva. Lysozyme has an immune function. It's an antimicrobial. So, obviously things make it through, right? I mean, we probably have all had some sort of a foodborne illness at some point. But lysozyme does stop a number of things from sort of making it through. So that's kind of a good thing. And then, of course, we have an enzyme in our saliva called salivary amylase, right? So if you guys remember from lecture this morning, we talked about the structure of starch, right? We said that it's this long chain of glucose molecules, right? So we have starch, right? And it's this long kind of chain of glucose with all of these branches on it, right? So it kind of looks something like this. You see, right? Kind of looks something like this, right? The starch is a very big molecule. It looks something like this with all of these glucose would be sort of lined up here. And we need to break it down. Amylase is an enzyme that works to do that. So what amylase does is it will come in, okay, right? Amylase will come in and it will start to cleave individual glucoses off of this. So we end up with little disaccharides. Okay? We end up with amylase. We end up with little disaccharides. Like this. But then the other thing that amylase can't do is it can't make it into these little branch points. These branch points are all just a little too tight for amylase. So we also end up with those little branch points that we're going to have to contend with a little bit later on as well. So you might end up with something that looks like this as well. Okay. So you might end up with something that looks like that because we can't quite get into those branches either. So we aren't completely breaking down starch into glucose, but we are doing an awful lot to start that process, right? We're getting little disaccharides, we're getting little things with two glucose linked together, and we're also getting these little things with these branch points in them as well. So we're definitely breaking it into small bits. You know, you start this process in your mouth when you mix your starch with your saliva. Of course, once you swallow your bite of food and that amylase gets into your stomach acid, it becomes denatured. So the process kind of stops for a little while, but at least we start the process here. But this can be, if you have the patience, this can be kind of a fun science experiment to do with kids, to teach them about enzymes and digestion. You know, you can actually, if you have the patience to continue to chew a bite of bread or something like that, over time you will break down that starch far enough that you do get these little disaccharides, and they'll actually bind to taste receptors on your tongue, and it will start to taste sweet over time. It takes like 5 or 6, 7 minutes for that to work, and most of us don't have the patience to chew food for that long. But if you keep the food in your mouth and you keep chewing, it will start to taste sweet after several minutes for that to occur. So it's kind of a fun science experiment with little kids because you can teach them about enzymes and digestion and the breakdown of your food. And it also buys you like 6 to 7 minutes of silence. And that's always a good thing too. So it can work out pretty well. So it's kind of a neat thing to do. That would be like the ultimate flex at Thanksgiving dinner, right? You just keep your food in your mouth and chew, and people say, "What are you doing?" I said, "I'm just letting the amylase work, man. Just letting it go." Probably not. Probably shouldn't do that. But nevertheless, it's an interesting thing. But it makes for a fun little science experiment because it's a super readily available enzyme. The other thing, well, let me show you guys some salivary glands here, and we'll talk about teeth. So your salivary glands are not on your mid-sagittal line. So on our digestive boards with our sagittal heads, we're not going to be able to see our salivary glands. So we need a couple of other models to be able to see those. We've got a couple of models for you guys to be able to see the salivary glands. So this is one of them right here. This is one of them. This is a head, but we're not on the sagittal line, so we can see them. You have 3, well, on each side you have 3 for a total of 6 salivary glands. The first one is right here. All right. This is probably the largest. It sits right over your masseter muscle in your cheek. Remember, your masseter is the one that you feel when you clench your jaw, right? It's the one right there. This is called your parotid gland. So this is your parotid gland that we see right here. A-R-O-T-I-D. And then the parotid gland has a duct that empties into the mouth right here. This one is pretty superficial. It's easy to get to. All you really have to do to be able to do it, you can actually squeeze your cheeks and you can actually eject saliva into your mouth. So just by pushing on this, you can actually eject saliva into your mouth. The other two are not as easy to get to. You have one underneath your tongue right here. This is called your sublingual gland. You see this right here. There are multiple very small ducts right underneath your tongue that the sublingual gland uses. Okay, so this is your sublingual, you see right here. Okay, your sublingual gland. And then finally, the one that we have right here, this is the submandibular gland. You'll see the mandible has been cut away right here for obvious reasons. And this one right here is right underneath the mandible. This is called the submandibular gland. So you have three, on each side, you have three salivary glands that secrete saliva into the mouth. The sublingual and submandibular and the parotid. It's kind of interesting. The submandibular gland, if I clear these labels, the submandibular gland's duct is right here. And it actually empties into the mouth right here, right behind your front teeth. These are your front teeth that you see right here. So kind of you're always bathing those front teeth on your bottom jaw. They're always kind of being bathed in saliva. When you go to the dentist to have your teeth cleaned, you probably note that that's one of the areas where they clean the most, right? Is that plaque that builds up on the back of those front teeth. I'm not judging anybody's dental habits or anything like that. I'm sure you all have very lovely mouths. It's an area where plaque tends to build up. A couple of reasons for that. One is that it's hard to get to, right? It's not the most convenient place for us to clean. But two, we're continually bathing it in saliva, right? The back of those front teeth are just continually bathed in saliva. And saliva is a great moist environment for bacteria to grow in, and then plaque builds up where those bacteria are dividing, right? So it's just an area that doesn't get cleaned very well and is just continually moist and everything else. So the next time you're at the dentist and you are getting the back of those front teeth scraped, just thank your submandibular glands for that, because that's who you have to thank. All right. Anyway, so just kind of an interesting aside. Let's take a really quick look at one more model that we have here. So this guy shows the glands, the salivary glands do really well. This one does as well. Right? So on this model, you guys saw this on the last test, right? Because we asked about the lingual tonsil that we see right here. But if we turn it on its side, we can again see the sublingual gland, the salivary gland, and we also see the submandibular gland. We don't see the parotid gland on this model, unfortunately, but we can see the other two. So this one also shows things fairly well, which I think is really good. The other thing, of course, that we can see on this model that I don't think any other model shows us is we can see our teeth, right? We have part of our jaw right here. And so what we can do is we can also look at the teeth, because our teeth, of course, are responsible for mechanical digestion, right? The breakdown of food into smaller pieces. So let's talk for just a second about your teeth. This is only a partial jaw. It's obvious the mandible's obviously been cut. It would come back a little farther. But it definitely gives us a really good picture of what our teeth do and the different jobs that they have. So let's start with these four teeth in the front, right? 1, 2, 3, 4. These four teeth in the front. When we look at them from this angle, we see that they're very, very flat on top. But when we look at them from this angle, we see that they're not very deep, so they're very, very sharp. Does anybody know what these teeth in the front are called, these four teeth? These are called your incisors, right? Your incisors. And boy, the edges of those sure are thin and sharp, right? What do you use something that's thin and sharp for?
Cutting.
Cutting, right? Yeah, use it for cutting. So, like carrot or something like that, right? You chop off a piece of that carrot, you use it to cut, right? Indeed, all right? So you have incisors on both the top and the bottom. You have 1, 2, 3, 4 incisors. Now, lateral to the incisors, you have this one and this one. Now, on this guy, and then most of us, these don't look a whole lot different. They don't look a whole lot different than the incisors, but they do have a little bit more of a point to them, right? We see a little bit more of a point right here, and a little bit more of a point right there, rounded if nothing else, okay? But still pretty sharp. What are these guys, the ones that are lateral to the incisors? Canines, yeah, these are your canines, right? And you and I, they aren't terribly developed, but if we look at other animals, like if you have a cat or a dog at home, we see that they can actually be really well developed, right? So if we look at an animal where they're very developed, like this mountain lion skull that we have right here, we see that these guys have really long and pointy canines. Ours aren't quite as dramatic as this, but this guy has really long and pointy canines. What do you think this guy uses these for? What would you use something that's long and pointy and sharp, right? It's not great for cutting, you don't use it to cut in the same way that you use your incisors. What would you use these for? Puncturing, right? And ripping and tearing, right? These guys obviously have a significant incisor because they probably also have to grab onto things and hold on to them as well, right? I mean, they have to subdue prey. But then they also will puncture and tear away bites of flesh as well, right? So these carnivores are using these really big canines to allow them to be able to do that, right? So we see that. We'll come back to this in just a minute, but we see enlarged canines. Ours are not nearly as enlarged, right? Ours are pretty boring compared, but they are there. Behind those, do you guys know what teeth are behind the canines? Called your premolars, okay? And then missing from this model, but behind those would be your molars, right? As a mammalogist, as a zoologist, I was always taught to just call them cheek teeth, right? Because they're back here where your cheeks are at. But they are the premolars and the molars, right? If we look at them, right, if we start to compare those to all of the other teeth that are in front of them, we look at them and we see that they have a much bigger surface area, right? That surface area that food is going to come into contact with is way bigger. It's way rounder than we see up here, right? These are just little edge that we're coming into contact with. This is a big area, right? You feel, if you use your tongue for a second, you just kind of feel your own molars and premolars, right? Right? They're a big surface area, right? But are they completely flat? No, they're not, right? They're like, they have some topography to them, right? Some bumps and valleys and stuff like that, right? So we can compare the cheek teeth of different types of animals as well, and we see what they do. What do you use your premolars and molars for? Do you use them to take bites? Not really, right? You use them to grind stuff up, right? Once you have your bite of food in your mouth, that's when you start to use it to grind things up, right? So let's take a look at the cheek teeth of different animals here too. I have a second skull with me today. Do we know who this came from? Do we have a guess on who this is?
That's a beaver.
What? It is a beaver. Yeah, good job. It's a beaver skull. Yeah. So beavers are kind of an extreme example of an herbivore, right? And I think we all know beavers because what do? They use these big incisors, these very what's the word I want to use? Pronounced incisors to cut down trees, right? They're really good at cutting down trees, and they can cut down a big tree, too. Don't kid yourself. They can get to work on pretty big trees. So we've got our incisors right here. Now the incisors of these guys, of most rodents, like beavers, are a little bit different. They're made of two materials. The front part of it is a really hard material, and then the back of the incisor is a much softer material. So you see that when the tooth wears, it wears at an angle like this. It makes it really, really sharp. And that's what allows them to cut down trees and stuff like that with this. The teeth also grow continuously. So this tooth goes all the way back up in the jaw, and it actually circles all the way back over to right about here. So if I were to pull this out, it would actually circle all the way through. And there's a lot of tooth in there still that needs to grow. It's pretty remarkable. So again, we use this to cut, right? And this is like an extreme example of a cutting tooth, right? That comes through and cuts plant material. Just like you use your incisors for, right? Let's compare that to the mountain lion's incisors, right? Remember, this guy's almost strictly a carnivore. And if we take a look at its incisors versus the beaver's, we see a pretty big difference there, don't we? These guys are puny, right? There's like nothing there, okay? Practically almost completely gone, right? But he has the canines to make up for it, right? What do we have canine-wise in our beaver? Oh look, they're not even there. They don't even have canines, right? There's no need for them to do that, right? They don't need to subdue prey, okay? They don't eat meat, at least not enough of it to justify canines. As far as the subduing of prey goes, a tree isn't going to go anywhere, so it's not going to run from them. So they don't need to do that. So they've actually over time lost their canines, and then they have a really big gap before you get all the way back to their cheek teeth, right? We said that these are for grinding, right? You said yours also aren't completely flat, but these guys, here, just feel this really quick, right? Is that flat? Pretty darn close, right? A lot flatter than yours, right? There's like no topography here at all. They're pretty darn flat. They're there really just to grind food, right? That's typically about what they do. Let's take a look at this guy real quick, right? This guy, it's a balancing act, right? They either sit on the shelf and never get touched, or they get touched and break. So I don't know which it is. But let's look at the cheek teeth on this guy. Look at those. I mean, even from this angle, you can see that they're really sharp. When we look at it like this, we see that they come up to an edge. They don't have that really flat characteristic that yours have and that the beavers have, right? Hang on. Get something else in here. There we go. All right. We get the bottom jaw of this guy. Right? The bottom jaw of this guy. Put it in here. One of the things that I want you to watch right here where my thumb is, watch the top tooth and then the bottom tooth that articulates it as I start to exercise the jaw. Look what happens when those two teeth come together, right? Look at what that shape is, right? It creates a little triangle right there and then chop, right? You have a lot of leverage in the back of your jaw, right? If you want to break open a jawbreaker, you don't try to do it with the front of your mouth, right? You put it way back here so you can bite with a lot of leverage, right? Don't do that. That's a good trip to the dentist. But if you wanted to do it, that's where you would do it, right? So back here in the back of the jaw, there's a lot of leverage, and look at what that creates. He's not using these to grind. What is he using these back teeth for? What do you think goes in there? Bones, yeah, they're using this to crush bones, right? So this guy's eating is breaking bones with this, right? These teeth are very sharp and they're meant to break bones, right? So it's a much different and a very specialized tooth arrangement than what you and I have, right? So we have two sort of extreme examples. cats are great examples of carnivores because of the way that their teeth are, right? And then we have our herbivore over here that also is a great example, exemplified here in this little rodent. And then we have us, right? We have our teeth. So I know they aren't as big, but whose incisors are ours most similar to? The most similar to the herbivore or to the carnivore? You think? I mean, again, I know they aren't as big, but like your incisors are pretty pronounced, right? Do you use your incisors a lot? Yeah, like every day, right? So I'm gonna argue with you. I'm gonna say that our incisors, even though they don't look the same, right? And we're not as buck teeth as a oh shoot, that poor tooth just broke. Oh no. It'll glue. It'll glue. I'll hold them in place. Even though they aren't as big, man, your incisors are pretty well pronounced, right? They're pretty big. All right, so as far as incisors go, we've got beaver incisors, all right? As far as canines go, what do we have? Well, the beaver doesn't have any. So the mountain lion wins, right? We've got mountain lion canines. And then what about the cheek teeth, the premolars and the molars? I'd almost venture to argue that we kind of have a mix of the two, right? Like, they're not perfectly flat like the beavers, but they're definitely not as sharp as the mountain lions. They're kind of in between, right? So if we've got an herbivore and we've got a carnivore, what are we? What's our dentition? An omnivore, right? We have an omnivore's dentition, right? And you may not be an omnivore, right? You might have chosen not to eat meat, but your dentition is definitely evolved as an omnivore's dentition. You have a little mix of what we see in herbivores, and you have a little mix of what we see in carnivores, kind of mixed together, right? If I think of it, I'll bring a bear skull down. We actually have a really similar dentition to that of bears, right? Bears are great examples of omnivores. They'll eat just about anything they come across. They will eat meat, but they eat a ton of vegetation as well. And they have a really good omnivore dentition. Obviously, the big difference between us and them is the canines. They've got massive canines because they can kill you. But beyond that, they've got a really good example of an omnivore's dentition. So you and I do have an omnivore's dentition, and we can see that when we compare what we have to these other animals. Kind of cool. All right. Gosh, this and snakes and everything else. You didn't know you were getting a comparative lecture today. All right. Any questions about teeth or saliva or anything? Yeah.
It's not really related to it, but like in terms of like bite force, is that due to leverage that the jaw creates, or is it because of the strength of the muscle?
Yes. It's probably both. The jaw does create a lot of leverage, so where the pivot point is and the lever will affect it, but also the strength of the muscle is another really important factor as well. Not all muscles are created equally. Some animals have muscles with just jam-packed full of actin and myosin, and as a result, they're a lot stronger than our muscles. That's why, like even with my dog, right? Like one of my dogs is only like 40 pounds or 50 pounds, but man, she is friggin' strong. And it's like, how is this little tiny animal so strong? I mean, my kids, when they were 50 pounds, weren't that strong. I could throw them around like rag dolls, right? But this dog is like strong. So why is it? It's because she's got a heavier sarcomere density in her muscles, and that makes her just simply stronger, right? The muscle per ounce is stronger than human muscle. But part of it is the way the jaw is built and where the pivot point is and stuff, and it becomes a leverage thing. So it's probably a mix of both. All right. So, Any questions? Any more questions about the teeth? That's a good question. Any more questions about the teeth or the saliva? No? Yeah.
So what is leaking caused from?
Leaking?
Yeah.
You're leveraging your sublingual glands. You lift your tongue up in your mouth and push forward, and you get those sublingual glands to eject saliva. Yeah, it's gross.
I can do it.
Huh?
I can do it.
I can do it too.
You? Yeah.
Yeah. Open your mouth, I'll show you. No, that's like the junior high thing, right? Like, hey, let me see inside your mouth. What? Ah, you just leaked in my mouth. Disgusting. You remember that in junior high? Oh. Yes, I was immature in junior high.
Oh, yeah.
I never really did it, but I knew not to open my mouth if somebody
It's
told me
all
to.
over the place, man. It's crazy.
But yeah, it's all sublingual glands. I wanted to point that out, but I didn't think anybody would get the reference, so I'm glad you brought it up. All right. So you chewed your food, you've broken it into smaller bits, you've mixed it with saliva at this point, and now you need to swallow it, right? We've started carbohydrate digestion, and now we need to send that bolus of food down towards the stomach. Swallowing is kind of a two-step process, okay? We don't normally think about it that way, but it is. The first thing that you have to do is you have to make the decision to swallow your food. You've chewed it to such a point where you feel like you're ready to swallow it. And what you do when you make that decision is you use your tongue and you push that bolus of food into your oropharynx. Okay? Once that food touches, well, once anything for that matter, touches your oropharynx, what it does is it triggers a spinal reflex that makes you swallow your food. Okay? So that's a reflexive thing. That's just a spinal reflex. I guess ostensibly you could learn to control, but it's just a reflex that sends that bolus of food down the esophagus. Okay? When you have that reflex, there's a sphincter. It's called the upper esophageal sphincter. Normally it's closed, preventing things from going down your esophagus. But when you swallow, what happens is that sphincter opens and it allows food to go down into the esophagus. Okay? And then after you swallow, the sphincter closes again. So this is why choking can occasionally happen. It's one of the causes. If you accidentally, when you're chewing a bite of food, push that bite of food back against your oropharynx, you can trigger that spinal reflex and swallow a little bit before you're ready. So that can sometimes go down your trachea, but even if an unchewed bite of food goes down your esophagus, it's a pretty uncomfortable experience. I think we've all probably done that from time to time, where you swallow before you're ready and you're like, oh, really hurt. And stuff like that. I have a funny story about swallowing, and the fact that it is a spinal reflex. So I have a friend that I grew up with. Friend is kind of a loose term. Do you guys have friends that like are fun to hang out with and make everything more fun, but they aren't like really there for you when you need them? Like, you're like, yeah, like I really could use some help today. Oh, sorry, busy, you know, but like they're really fun to hang out with. You know, like they're kind of like, you know, not real friends, but they're fun to hang out with. If your group of friends doesn't have somebody like that, you are that person. I'm just kidding, that's not true. But at any rate, I had this friend named Chris who was like that. And man, the guy was hilarious, but never there for you. Anyway, he tells this story that's hilarious. So he has a little brother who's like 2 years younger than you are. He was the sassy one. I guess it's always the younger brother. When he was like 6, he wasn't allowed to play with the older neighborhood kids because of his mouth. His mom was like, yeah, no, you're corrupting everybody in the neighborhood. You got to tone it down. Then he would say something Bart Simpson-esque and then go on his life. But at any rate, he tells the story about him and his little brother. And I guess he must have been around 6 and his little brother was around 4 or something like that. And their chore when they were younger was they had to clean out the cat litter box. their mom would save the produce bags from the grocery store for that. You know, the little plastic bags you put like vitamins and stuff in at the grocery store. She'd save all of those. And so they're cleaning it out and stuff like that. I don't know, mom was like across the street at the neighbor's house or something like that. So they finished the chore, and my friend is like got the bag of cat poop in his hand. He's twirling it around like this. And his little brother says, dude, stop it. The bag's going to break. Shit's going to go everywhere. And he's, oh, shut up. It is not. Well, at that moment, the bag broke. And a cat turd flies across the room. And his little brother is standing there like this. The cat turd goes right into his mouth. But here's why I'm telling the story. It hits his oropharynx and triggers the swallowing reflex. And the cat turd just goes right down the hatch into his stomach. And there you have it. I guess by the time his mom got home, he convinced his little brother that he only had hours to live. And that he was definitely going to die. Obviously he didn't, but he convinced him that he was going to. So there you go. It's an involuntary reflex. Nobody would ever voluntarily swallow a cat turd. So it just goes to show you that it's an involuntary spinal reflex that when something touches the back of the oropharynx right here, that you reflexively swallow. All right. So not much happens in the esophagus. Obviously, the process peristalsis is what's moving the bolus of food down to the stomach. So we have that kind of cooperative action between the circular and the longitudinal layers of muscle in the esophagus, and then we get to the stomach. Right? Just a couple of quick words about the stomach. We spent quite a bit of time on Friday of last week looking at the anatomy of the stomach, so I won't bore you guys with it again, but just a couple of quick review things. The first is that it's important to remember that you can fill your stomach pretty quick, right? Like you can eat a meal fast if you have to. I can put away a burrito in just a couple of minutes if I have to, right? But we can't empty the stomach as fast as we can fill it, right? If we did, we wouldn't be able to get the most nutrients out of it, right? So even though we can fill our stomach relatively quickly, we have to really control how quickly we empty the stomach into the small intestine. Okay. So once the stomach starts to distend, once we start to put food in it, it triggers this sort of chain reaction that causes us to really control how fast we are emptying. The vagus nerve is obviously playing a big role here, but there's a whole bunch of things that are going to be secreted as a result of this. I'll go over a couple of them right now. But this is where the control of motility really starts, is in this gastric phase of digestion. Okay. So last week we talked a little bit about hydrochloric acid and intrinsic factor that are secreted by the parietal cells. We'll review those in just a moment. We also talked about pepsin and gastric lipase that are also secreted by the chief cells of the stomach. And we looked at those under the microscope on Friday. A couple of cells that we didn't mention on Friday were the G cells and the D cells. The G cells secrete a hormone called gastrin, and the D cells secrete something called somatostatin. Both of these are hormones that are going to really regulate the rate of gastric emptying. So again, how fast are we secreting things into the small intestine? Again, even though we fill the stomach quickly, we have to really slow down and only eject small amounts of material into the small intestine at once. So it's a very, very slow process. So really starting to regulate the rate of motility once the food starts to enter the stomach. Let's take a really quick review of the parietal cells and the chief cells and look at what they secrete. So remember that in the mouth, we mixed our food with saliva, which has salivary amylase with it, and that started to break down starch. We haven't actually broken any of our carbohydrates down to monosaccharides yet, but we definitely have started that process. in the stomach, we start everything else. So remember that I said this morning in lecture that proteins, right, have this chain of amino acids for proteins. This chain of amino acids, right? But usually those proteins are folded into fairly complex three-dimensional shapes, right? And that three-dimensional shape is really, really important for helping the protein to do its job. And we said this morning in lecture that one of our big challenges is that we've got to unfold this in order to be able to break it down. When we unfold a protein or cause a protein to become unfolded, what's the name that we use for that? It's denaturing, right? So we need to denature the protein, right? What are a couple of things that we can what are a couple of things that could cause a protein to become denatured? Temperature is one, right? Putting on the heat is one thing. What else? pH, right? Those are two of the important things. There are other things that can cause proteins to become denatured, but temperature and pH are two of the most common things. Temperature is an interesting one. It's actually really easy to use temperature to watch a protein denature in front of your eyes. This is another fun thing to do with kids and also teaches them a good life skill. Fry an egg, okay? If you want to teach kids about the way enzymes work and the way proteins work, or at least the way proteins work, fry an egg. And you crack a raw egg open, the egg white isn't white, right? It's clear. But as soon as you put it in the frying pan and expose it to heat, it turns white. That's because of the protein in the egg white starts to denature, it starts to change shape when you expose it to heat. And when it changes shape, it reflects light differently, and we see that color change with our eyes. Right? So when that egg white turns white when we cook it, that's because the proteins are becoming denatured. This whole thing is like a lab for kids, right? We've got salivary amylase, we can denature proteins. Let's see what else we can do. And we can get a bunch of 6-year-olds in here. They won't have any questions about that. They're going to ask other random questions. But yeah, denaturing, right? So temperature is a great way to denature a protein. The problem is there's no real way for me to expose the protein that I eat to 140 degree temperature, right? And what we call it endotherm. I maintain a relatively constant body temperature. And as a result of that, the inside of my stomach is 98.6 degrees, just like the rest of my body, right? So exposing it to heat isn't really a conceivable thing, but exposing it to a change in pH is, right? And that's why our hydrochloric acid that we produce in our stomach works to denature our proteins, right? So the hydrochloric acid denatures the protein, right? So proteins are, denatured by HCl, right? HCl, the hydrochloric acid that we secrete is going to denature that protein, and it's going to take it from a three-dimensional shape into a primary structure, a string of amino acids. Okay? So the hydrochloric acid denatures the proteins. This is like strong acid. We're talking like inside of your stomach can have a pH as low as 1. All right? So like, we're talking battery acid acidity here. Right? This is super corrosive. Your stomach secretes a lot of mucus to help to protect it from that acid. But if the mucus fails, you can actually start to digest the epithelial cells and everything else in your own stomach. What's that called when that happens? When you start to digest a hole in your stomach? called an ulcer. Right? When you get ulcers, we're actually digesting where that hydrochloric acid is corroding and wearing away the actual lining of your stomach, right? So we definitely want to avoid that. All right. The other thing that we do secrete from our cheek cells is we secrete that enzyme pepsin. Pepsin is almost immediately converted to pepsinogen, which is why I put it in parentheses. But pepsin, okay, pepsin is an enzyme secreted by the Chief cells, and pepsin belongs to a family of enzymes called endopeptidases. Belongs to a family of enzymes called endopeptidases. I'm not so concerned that you remember that vocabulary word, but the prefix is really important, right? What does that prefix endo mean? It means inside, right? We've seen it all semester, right? The endothelium of our blood vessels, the endo, all the endos we've had this semester. I'm blanking on all of them. Right? But we've seen it all semester long, right? So endopeptidases can break apart amino acid chains, but for whatever reason, and I don't know why, they can't go to the end of an amino acid chain and cleave off individual amino acids. They can only work on the inside of the amino acid chain. So pepsin, as an endopeptidase, can do this, and we have a shorter chain of amino acids, but it can't cleave off individual amino acids. Does that make sense? So we can do this, we can do that. So we're cutting that amino acid into smaller pieces, but we aren't yet able to completely break it down into individual amino acids. There's a whole other family of enzymes. If we have endopeptidases, what do you think the other one is? Opposite of endo is exo, right? So we're going to have exopeptidases later on that are actually going to be able to cleave off individual amino acids. But pepsin happens to be an endopeptidase. So a couple of things at play in the stomach. We're starting protein digestion. We're denaturing the proteins with hydrochloric acid, and then pepsin is working to help to start to break them down. But still, we don't have individual amino acids yet, right? We just have smaller chunks of amino acids, right? Some of these proteins are pretty big. Like we're looking at 2, 3, maybe 400 amino acids that are linked together. So just chopping them to smaller bits is kind of what we're doing. But we don't have the individual building blocks. Okay, so we have hydrochloric acid and pepsin working together to help us to break down proteins. It's worth noting that hydrochloric acid also kind of has an immune function as well. Just about anything that comes into contact with that acid gets destroyed, right? So bacteria, things like that are definitely less likely to survive once they come into contact with the really acidic environment inside of the stomach. And there are certain types of things that can form cysts and protect themselves from the acid environment in the stomach, but a lot of things are susceptible to and then finally we have this enzyme gastric lipase. Gastric lipase works to break down fats. Remember, the structure of a fat is we have a glycerol molecule, and then we have 1, 2, 3 fatty acid chains attached to it. Lipases work by going in and cleaving off individual fatty acids, right? So lipases kind of come in, and then they work by cleaving off individual lipase. They'll cleave off individual fatty acids to make the molecule a little bit smaller. Right? We'll talk a little bit more about fats later on, but we are starting. Excuse me. We are starting the process of fat digestion here as well. So to recap, right, in the mouth, we started carbohydrate breakdown. In the stomach, we start fat breakdown. We also start protein breakdown. But none of it is finished at this point, okay? I think, going back to our little kid example, right? If you were to go up to a little kid and say, what does the stomach do? I think we all sort of grew up thinking that the stomach digests our food, right? That's kind of the way that we always perceived it. I know I did. But the reality is that we aren't really doing anything other than storing food in our stomach. We are breaking it down a little bit, but really the stomach is a storage organ, right? Again, we fill it very, very quickly, but we empty it, we can fill it in just a few minutes, but we empty it over several hours, okay? So the stomach is a storage organ that regulates how fast everything makes it to the rest of the system, okay? So it's definitely a storage organ, even though there is a small amount of digestion. So again, I know we went over this on Friday. Any questions about the stomach? All right. So the stomach also has a little bit of mechanical digestion occurring. It's really mixing and churning your food. It's a very muscular organ. We saw that on Friday. Not only do we have the circular and the longitudinal layers of muscle, but we also have the oblique layer. So it's really doing a lot of churning and mixing of your food and breaking it down. So there's a small amount of mechanical digestion that goes on there as well. The liquid that is pushed to the small intestine is called chyme. It resembles kind of a heavy cream, but it actually gets injected into the small intestine. So let's take a look at your small intestine and talk a little bit about it. So the small intestine is divided into 3 regions. It's a very long organ. It's about 22 feet long. And this morning, we put a small intestine on the floor to sort of represent it. I started the small intestine over here with a piece of tape, and the small intestine at about 22 feet long goes all the way over to about right here. That's where we guesstimated about 22 feet is. So this is our small intestine right here. It's fairly large. It is divided into 3 regions. The first 5% or so is the duodenum. So from right here, right about shoulder width apart, if I stand right here, this is my duodenum. I actually wrote duodenum on the tape here so you can see it. But then the other 2 sections, the jejunum and the ileum, are roughly equal in terms of their length, right? So they make up the rest of it. So, if I'm starting at the wall, the duodenum ends right here, but then I have to go a little ways. I go all the way through the jejunum, which ends about right here, and then I go all the way to the end of the ileum before I go into the large intestine, which would be over here, right? So we have that. So really, really big organ, okay? And a lot is happening here. It's worth pointing out that because it is such a big organ and there's so much that needs to be done, those 3 sections are actually really, really important, okay? The same thing doesn't happen in each section. And the anatomy of the small intestine changes a lot as we go through, okay? So we have a model of the small intestine. Put this up really quickly. We do have a model of the small intestine. And there's some key features here, but they definitely aren't all features, right? So this is our model of the small intestine. It's kind of similar to the stomach that we used on Friday. It shows the main layers, right? So we'll look at this under the microscope in a little while as well. You've got your mucosa, you have your submucosa, you have your muscularis externa, and then you have your serosa here. On this model, what they're doing is they're trying to show everything that you would find in the entire small intestine in literally 6 inches, right? So it's great to be able to see it, but just know that you're not going to see every single thing that's on this model and under one microscope slot. We're going to have to move around a little bit. A couple of the notable things that you're not going to see everywhere is this guy. You'll notice that you have this structure that starts in the submucosa and extends into the mucosa. This is a lymphatic nodule. It's called a Peyer's patch. These guys are kind of like tonsils. Anytime you bring something into your body, you have to sort of monitor it for infection happening. We're going to see these guys until we're in the ileum, out here at the very end of our piece of tape, right? Because that's when we're going to be absorbing things, and we'll talk a little bit more about that. The other thing that we see is that we see in the submucosa is this really notable gland. It's called a submucosal gland, which is descriptive. But what it does is it secretes bicarbonate into the duodenum, right? Remember that when that chyme comes from the stomach, which is over here, right? When that material comes from the stomach and it comes into the duodenum, it's still super acidic, right? So what we need to do first is we need to neutralize that stomach acid. And the way that we do that is by secreting bicarbonate from those submucosal glands. So when we look at the duodenum under the microscope, we're going to see a lot of those submucosal glands. But as we move down, the glands become less and less and less common until they disappear entirely. But again, once we're in the ileum and we start to absorb nutrients into our bloodstream, that's where we see these Peyer's patches starting to show up, and we're going to see those a lot more down here. Okay? So again, the anatomy is not going to be completely the same. Some things that are the same, though, is the fact that the small intestine is folded in a very similar way to the way the stomach is. On a big macroscopic scale, we've got our plicae that we looked at last week on our digestive warts. Pull up the other small intestine here in just a minute and show you the plicae. The plicae. But on a microscopic scale, you see that the membrane is even more folded. These are villi. Okay. So we have villi. I guess they're sort of analogous to the gastric pits. These big folds that we have here, these are villi, these finger-like projections. I guess the invaginations of those folds that we see right here were what we call gastric glands in the stomach. These are called intestinal crypts. Okay? Intestinal crypts. You guys know what a crypt is?
No, like the crypt keeper? No?
So these are called intestinal crypts that we see here, right? So this is an intestinal crypt, which is glandular, okay? It does have a lot of secretions that we'll talk about, but it's a glandular structure, right? So we have villi and we have crypts. Crypts. Not crypts, those are different things. Crypts versus blood, that's beyond the scope of this class. So here you've got your your epithelial cells, right? We do have a couple of goblet cells that are thrown in here. Those are these little splotches of paint meant to represent a lot of mucus being secreted. In the duodenum, way over there, that mucus protects the lining of the stomach from acid before it becomes neutralized. But all along the length of the small intestine, it actually helps to lubricate our food and stuff like that. And sort of keep it moving, right? So we have that. On top of that, we see that the individual epithelial cells, so the epithelial cell is right here. So this is one epithelial cell that you see right here. There's another one, all along here. These are all epithelial cells. The epithelial cells have microvilli that we can't even see on this model. These folds in the plasma membrane, right? So what we're doing is we're increasing the surface area of these cells considerably by folding the plasma membrane along here as well. Those are called microvilli, right? So what we're left with is we're left with a massive amount of surface area, right? huge amount of surface area in the small intestine, right? So this is my small intestine. Imagine that I've got this little tube, probably no wider than the width of this piece of tape going along. Right? Let's take a scalpel and let's take it and cut it open so that it lies out flat, right? Let's do that really quick. Right? Let's take a scalpel and let's lay it open. So if I did that, if I could cut this tube like I cut a garden all the way down its length and laid it out flat, right? So we increase the surface area a little bit, right? Right? Like that. Don't mind me, I'm just taping. All right, it would be like that, right? Gotta go get on the other side, hang on. Don't go too far. Okay, then I come down here, same thing. There we go. So now, our small intestine is laid out pretty flat, right? See how much bigger it got, right? Now it's not the width of the tape. Now it got a little wider because we laid it out flat, right? Okay, but now we have to use our imagination. Now we need to get out an iron of some sort, right? Because we need to get all these wrinkles out, right? All those plicae in the small intestine. What I want to do now is I want to take those and I want to iron them out so they're flat. So let's do that. When I do that, my small intestine is not only going to get wider, it's also going to get a lot longer, right? Because I'm taking those wrinkles and I'm making them go out, right? So now, when I'm done with that, my small intestine is going to be about this long, right? And it's going to be a lot wider, right? So it's going to be like this. I'm not going to go all the way. Maybe I will. Right? It's going to be like this. So by the time I get rid of those plicae, I see that my small intestine is in this giant box that goes all the way back. All the way back. Isn't that a fart? No, here, we're going to make it even bigger, right? And this is just the plicae. Okay, so now, look at how much bigger the surface area of your small intestine just got. But now, I want to take that same iron, and I want to take the villi, and I want to iron them out as well, right? That was just the plicae. But now, let's take all those millions of villi that are in there, and let's empty those out as well. When I do that, you're going to have to move. Get out of my way. When I do that, now, my small intestine is ironed out like this. Right? Now it's ironed out like this. I'm not going to go all the way back to the wall. But I am going to do this to give you an idea of how big a small intestine is. Right? There. See how much bigger that gets? Now we have microvilli that we have to iron out. So let's do that. I'll be back. I gotta go to the microbiology lab.
And this belt has little trousers.
I'm just kidding. I'm not really gonna go put tape on the floor in the microbiology lab. But it would go all the way over across the hall, all the way into the microbiology lab. And it would come all the way over, like right here, right? And it would go all the way into the 50A lab next door. See how big it is? That's a huge surface area compressed into a very, very small space. Crazy, right? All right, there we go. There's our gut. There's our small intestine. Huge, huge surface area compressed into a really, really small space. I should leave this for Ari. What are you doing here? I'll be 17 minutes. There's like a Ouija board in there. Judith never walked in this lab again. By now, this shouldn't be a foreign concept to us, right? Our body is really good at fitting massive surface areas into very small spaces. We saw this with the lungs and our alveoli. We saw it when we studied the circulatory system and our capillaries, right? Huge surface areas compressed into very, very small spaces. If there's anything our bodies are good at, it's that, right? So we can see that when we take a look at this. Okay? All right. So the anatomy of the small intestine is such that we do see this all the way through, right? There are some things in the small intestine that are localized to the duodenum or the ileum, but there are some things that we see throughout. All right. Let's go ahead and take a look at the small intestine under the microscope. The other day, I asked you guys to grab a slide. The digestive composite. If you just come up and grab that same slide that we had on Friday, let's take a look at the small intestine on that. I'll throw it back up here so you can kind of see what we're Alright, so you should be seeing something that looks similar to what I have up on the board right now. You should see something like this. If you're not seeing that, I can find it out. Okay, so I like this one a little bit better. So this is pretty boring small intestine right here. It has all the basic layers and has all of the stuff that, or has the villi that we've been talking about and stuff like that. But it doesn't have any of the glands or anything else. So probably we're looking at a section of jejunum right here. We're probably somewhere right in the dead center, right in this area of our small intestine. Right? We're not in the ileum, we're not in the duodenum, we're probably squarely in the jejunum, kind of right in here. Right? So take a look at this. I'm going to ask you to try to identify your 4 layers of your small intestine. Right? So try to identify the mucosa, the submucosa, the muscularis externa, and then I can also see the serosa on here as well. I have a little bit of serosa on here as well. So try to identify your 4 layers: the mucosa, the submucosa, the muscularis externa, and the serosa. We'll talk about it in just a minute. We'll see how you did. I want you to try to find it. Try to figure it out first. Talk to each other about it. It's not a test. guys are looking, I'm gonna go refill my water. I'll be right back. My voice is going today. I should have gone into the micro lab. Dr. Lisa would have gotten really upset with me.
You have a good reason to.
Huh?
You have a good reason. Good reason to.
I know, right? It's demonstrated.
Go to the fish player. Or if we just
How are we doing? Do we have success? This is a beautiful layer cake. So, where are we at? How are we doing? Figure it out? You guys get a label? See what your layers are? Alright, see how we did, okay? So obviously this is the lumen of the small intestine kind of right here, okay? The edge of the lumen is over here, so the mucosa starts there. And then ends right about here, right? This is the end of our mucosa right here. So the mucosa, this layer kind of right in here, right? Submucosa is all of the connective tissue that we see right in here. So this is the submucosa that we see right in here. And then you have muscularis externa. You have a very distinct layer of smooth muscle right here. This is your muscularis externa right here. And then you have a little bit of serosa up here, and a little bit of serosa right here. So how do we do? right. All right. Go ahead and zoom in on anything. Those are your basic layers of your small intestine. Really nice thing about the small intestine is it's way easier, I think, than the stomach is. You fix your labeling if you need to, and then we'll kind of go on from there. Alright, so what I would like to do is I would like to zoom in on the mucosa and look at just a couple of features of it. I'm going to clear my marks and go ahead and we can zoom in on the mucosa a little bit and kind of see what we have here. So those villi are going to be really obvious, right? They're these finger-like extensions that, at least in my case, are pointing to the left, right? Those are the villi. Hello.
Came to see your intestine.
Came to see the intestine all taken out.
Yeah.
So that last piece of tape is only showing the width. It actually should be in the micro lab across the way, and it's got to go into the 50A lab in the process.
Yeah.
So the
Drill a
green,
hole in the wall,
so the
or-
green tape. No. Bear with me for a minute.
Okay.
So the green tape is the small intestine, just the tube
Okay.
that you take out, and then
Okay.
the pink on either side of it is like when you cut it open and lay it out flat.
Okay.
And then the plique, the big like folds that you can see with your naked eye, would be inside of this box. So once you iron them out flat, it's like this.
Okay.
And then the villi, right? It would be a box like this big, like right here. So you're in the villi right now.
Okay.
Okay? And then the microvilli, once we do that, they'd be like way wider. They'd go from like that wall all the way over into the 50A lab, but then it would actually go all the way into the micro lab.
I love that. So we can break all the walls down so we can.
Can we?
We have a
Let's go out to
sledgehammer.
let's go out to the parking let's go out to the parking lot.
Yeah.
Take a field trip. Anyway,
Tell them to bulldoze like all the new parking lots so we can like make this.
make
out there.
this. I'm gonna
Yeah.
paint it on there. This is your small intestine. That'd be amazing.
Paint it on the floor here. Okay, good. I'm
You
glad.
don't, I know you don't care.
I'm glad I
I won't
inspired
get in
you.
trouble.
Yeah, no, you did. So
Good.
yeah, I
I
appreciate
don't
the
even
idea of
recognize
paint on
these
the
tape
floor.
covers.
Huh?
What's the washi tape?
I just used the masking tape
Or
that's
no,
in
the
here,
masking
so
tape. Oh.
yeah.
Good. How exciting.
Yeah. All right. I was telling the lab techs about our invention. I feel like this is frozen over there. All right, let's go ahead and kind of move on here. So the villi are these finger-like extensions, of course, and then the epithelial cells would be around the edge of those. So let's zoom in on those real quick. Let's take a look here. So chances are you're not really going to have the ability here to see the actual microvilli. They're just too small to see, and these slides aren't really good enough. But you can definitely see the epithelial cells, right? All of these columnar epithelial cells that make this up, and then you've got these guys. What are these big cells that you see here? Those are goblet cells. Yeah, those are where we're secreting mucus from, right? So in the mucosa, we can definitely see the epithelial cells. We can see the goblet cells. Unfortunately, we just can't really see the microvilli, which is kind of a shame. And so take a minute, let's take a look at those. Take a look at the goblet cells and stuff like that. There's one other thing I want to point out in the mucosa, and then we'll sort of move on to other parts of the small intestine. did want to point out, and I forgot to mention it a couple of minutes ago, and I apologize, is that you notice that each of the villi has a blood supply to it. So you'll notice that you have a little artery and a little vein coming out. So there's a little capillary bed. Each one of your villi has those, right? But the other thing that you'll notice is this green vessel in here as well. This is a lymphatic vessel that we see here. These lymphatic vessels, they're called lacteals. This is called a lacteal. Okay. That lacteal plays a really important role during the absorption process. So we'll return to those guys a little bit later, but that lymphatic vessel is really, really big. And every single one of these villi has one, right? And we're talking about literally millions upon millions of villi, right? But every single villus has a lacteal in it, and that's going to be important a little bit later on. To see those lacteals under the microscope, the easiest way to kind of do that, zoom out a little bit. One of the things you see, like in this one right here, and that one below it, both of them. You notice that you've got a hollow space right here. That's the lacteal, okay? It is a continuous tube that runs up the middle of the villus, but what it's probably doing, right, is, it's right here. What it's probably doing is the lacteal is actually like out here, away from our plane of view, and it's going and going and going, and then it turns and it goes in there. And that's why we see it right there, but we don't see it anywhere else. So that little opening, well, that big opening, I should say, is a lacteal, and it's kind of running through the middle of the villus, and you can see them. And you might end up with a slide where you can see the whole lacteal in cross section going all the way up the villus, but that's really rare to be able to see those. So this is what you're more likely to see. Let's see if it's full one from my slide. You might have to be luckier with your own, but I don't see the full one. Anyway, take a look at those. I'm not terribly likely to ask about lacteals under the microscope. Much more likely to use the model. But I just wanted to show this to you. The other thing that's noteworthy, or that I'd like you to take a look at, I should say, on this is if you go back a little bit, you go through the submucosa, you get to the muscularis externa, you're going to see that in the small intestine, we have the two layers of muscle. We have the inner circular layer, and then we have the outer longitudinal layer. These ones are really easy to find because they're at like perfect 90-degree angles to each other. That inner circular layer is way thicker, but you can see where those muscle fibers change direction. So identify those muscular layers here, the circular and the longitudinal layers, and then we'll move on from there. So we all good with the basic? Oops. We all. All right. All right. Let's just go ahead and do this instead then. So are we all good with the basic anatomy of the small intestine? All right. Well, let's go ahead and talk about the different regions then, right? What happens in each region? Because again, with something this large, there's a lot happening here. Remember, we haven't even started or finished. We haven't even finished digesting. Right? We started carbohydrates, protein, we started fat. Nothing is done. So we have to finish digesting our food, and then we also have to absorb all along this 22 feet of small intestine. So in the duodenum, right, and we're right at the very beginning here, the primary goal here is to do a lot of secretion to help us digest our food, but we also have to protect the small intestine. Right? Remember that the digestive coming from the stomach, which is behind me over here, is really low pH. This, as we mentioned, is incredibly corrosive. So we have to secrete things into the duodenum to allow us to protect ourselves. The duodenal glands or the submucosal glands are going to secrete the bicarbonate. That's going to be the primary thing that is going to neutralize stomach acid. So one of the things that we're going to see when we look at the duodenum under the microscope in just a couple of minutes is we're going to see these duodenal glands or submucosal glands under the microscope. They go by a couple of different names, right? This is a gland that has a duct that bicarbonate travels to the lumen through, and that neutralizes stomach acid. Okay? So the submucosa of the duodenum is just almost exclusively those glands. Okay? The other thing that we have to do is we have to secrete a lot of other digestive enzymes in there so that we can finish digesting our meal or our food. And the duodenum is directly connected to things like the pancreas and the liver, right? So if we look at this model right here, and if you take a look at this model right here, this is this little section of the duodenum right here. The stomach, of course, would be up here and then continue from there. But you'll notice that the pancreas is right here. There's this duct that the pancreatic enzymes are going to flow down, and they're going to empty into the duodenum right here, and then through that secondary duct right here. We talked about these a little bit prior. So there's a lot of secretion from the pancreas happening here. The liver is also sending a substance called bile, which we'll talk about probably on Friday, but it's sending a substance called bile into the small intestine through this duct that you see right here called the common bile. All of that's emptying into the duodenum. And then, of course, the duodenum itself is also making digestive enzymes that are going to be secreted. Right? So there's just a huge amount of secretion that's happening. We do want to avoid any absorption. So to do that, we actually are secreting an isotonic sodium chloride solution in as well. What that does is it helps lubricate the food, but it also avoids creating an osmotic gradient, which would lead to fluid loss. So the isotonic sodium chloride solution just keeps water in the gut, at least for the time. So a number of things being secreted in the duodenum of the small intestine. The name of the game for this first foot or 2 feet of the small intestine is secretion. A lot of secretion happening. In fact, we probably should write that on our tape so that we remember that, right? I just labeled it for duodenum. All right. Anyway, so we have secretion happening there. All right. All right. Let's take a look at the duodenum under the microscope. In just a moment, I'm going to ask you guys to come get a second slide. This is the stomach and intestine slide. Let me show you what we're going to be looking at really quick, and then I'll ask you to get that slide. So, we've got this small. You guys looked at this on Friday. You guys spent some time with this, right? Your stomach is right here. You guys spent some time with this. Obviously, food is coming from the esophagus here. It's coming through, and then it goes through the pyloric region of the stomach, the small intestine right here. What's that little muscle that sort of holds this end of the stomach closed called? It's the pyloric sphincter, right? It's the pyloric sphincter. So this little area that we see right here is the junction between the stomach and then the duodenum, which is right here. The duodenum literally starts right here. Okay? Under the microscope, this is what we're going to be looking at. Okay? See how similar those two things are to each other? Yeah. Right? So this is literally the end of the stomach right here and the beginning of the small intestine right here. Keep in mind that under the microscope, it's all going to be upside down, right? So the stomach is going to be on top, and then we're going to go down into the small intestine. But what we're going to be interested in here is we're going to be interested in the very beginning of the small intestine, and we're going to be looking at the submucosa, because this whole part of the small intestine right here is just jam-packed full of those submucosal glands so that we can neutralize the stomach acid. Right? So let's grab that slide, let's throw this one on the microscope, and let's see what we can see. The small intestine. If I scroll over just a little bit, you'll see that pyloric sphincter right here, that really thick layer of circular muscle. That's really all a sphincter is, right? It's a circular muscle that closes the tube off. So it's just an enlarged area of circular muscle. The circular muscle layer is already there, okay? But it's just really big when we run into the sphincter, and that allows us to close the tube off. So what you do is you scroll down just a little bit, and you see how that sphincter, that circular layer of muscle, comes back to a normal width when we come in. But nevertheless, this is now we're in small intestine. So what we want to do is we want to find an area of the small intestine. Anywhere will work. It doesn't have to be perfect. We're also in a really funky area of the small intestine. Like it's literally the junction between the stomach and the small intestine. So things aren't going to look as pretty as they just did on the slide that we just took off of the microscope. And I don't expect them to be, I don't expect you to be able to interpret them. The only thing that I care about on this slide is the submucosa, okay? That's the only thing I care about on this slide. So let's come over, let's come over in here, and let's find that submucosa. So, this is our muscularis externa right here. This is our mucosa right here. So that makes all of this our submucosa. Let's go ahead and take a look at that area. Again, it looks really funny. It's not as clean as what we just did. But we go past that mucosa, and then some of it we get in cross section, some of it we get in longitudinal section like this, but it's all in there, right? All we have to do is just kind of use our imaginations, and we see that all of this is that submucosal gland. And it's just all it is, right? Just end to end submucosal gland. We have to neutralize it. So we look in the submucosa, we're in this region of the small intestine. That's what we're going to find. Right? So take a minute to identify the submucosal or the duodenal glands. Submucosal glands. Beautiful. Nobody can neutralize acid like you. You go, champ. That's all of them, yeah. Where's the sun? Yeah, I don't know. I don't know if that's a slide trap or what that is. Yeah, I don't really know what those are. That's a good question. All right. Submucosal glands. I guess I can hook up some of my tape. It's funny, I know exactly where people stepped. I went from a shoe print to the tape actually stuck to the floor. If people didn't step, it just pops right off, it's not stuck, but it's really stuck where people step. Alright. How are we doing with our submucosal glands? Finding them? They're kind of cool, right? But, I mean, they're so dense, right? They're just one continuous gland. Yeah, what we're doing here is we're secreting a whole bunch of stuff into the small intestine. The bicarbonate from these submucosal glands are only the beginning of the story. Right? We've got a whole bunch of other stuff that's being secreted. So let's go back to our list really quick. Let's just take another look here at what we've got. See if PowerPoint comes up for this time. All right. So let's take a quick look here. So the bicarbonate, we just looked at those glands. We've seen goblet cells. We know that those exist. They're secreting the mucus. Let's talk a little bit about the digestive enzymes. So the digestive enzymes are kind of the exception here. We are obviously secreting the pancreatic enzymes into the duodenum. There's a lot of pancreas secretions here. The bile that is coming from the liver is also being secreted in here. But the digestive enzymes don't just end there. Even in the beginning of the jejunum, if you start to walk down, you see that we have enzymes that are being secreted by the small intestine itself, right? So far as the pancreas goes, let's go ahead and add some notes here. As far as the pancreas goes, right? As far as the pancreas goes, the pancreas, like the salivary glands, also secrete an amylase, right? So we have pancreatic amylase as well. So we have amylase that's being secreted. The pancreas also secretes a lipase as well. Okay, so we have amylase, we have lipase. That's being secreted by the pancreas. This morning in lecture, we talked a little bit about our sugars at this point, right? A lot of them exist as disaccharides. Right? We've got the lactose, maltose, sucrose, and stuff like that, right? So we kind of lump those into a category of enzymes that we call disaccharadases. Lump them into a category that we call the disaccharadases. It does include things like lactose and lactase and maltase, sucrase, and all of these enzymes that break down these really common dietary disaccharides. So we just call them disaccharidases. That way we lump them into one thing instead of having to list them out. So we have those, okay, that are being secreted. We also need to be able to break down our amino acid chain. We need to be able to break them down into individual amino acids. So we're going to need exopeptidases as well, right? So we've got a whole host of peptidases that are being secreted that are going to, both endo and exo, that are going to be important as well, right? So we have peptidases that are being secreted as well. So your pancreas really works hard to make sure that you're secreting a ton of enzymes at this point, right? Amylase, lipase, saccharadases, peptidases. We're definitely not going to spend the time to learn the names of all of them. I can do that for you, but we don't want to learn every single enzyme that's secreted. I want you to understand, though, that we're kind of getting to this point where we need to start getting these food molecules into their individual building blocks, right? So we're giving our small intestine the tools that it needs to be able to do that. Okay. So lots of stuff being secreted into the duodenum. Most of it's coming from the pancreas, right? Now, the small intestine will secrete enzymes into the small intestine throughout, though, especially into the jejunum. Real quick, just let me show you what those look like, and we'll talk a little bit about some other stuff as well. Let me come back over to the So the small, in addition to the pancreas secreting enzymes, the small intestine will actually also secrete enzymes. So you'll see little enzymes being secreted here by the small intestine into the lumen of the small intestine. Okay, so this would be your three-dimensional enzyme that you have right there. Problem with secreting these enzymes into the lumen of the small intestine is that they get washed away, right? This is all moving fluid through here. So if I secrete them in here, they tend to get washed away. So the enzymes, a lot of the enzymes that are secreted by the small intestine have a really unique feature. And what they are is they're actually going to be tethered to the wall of the small intestine. So there's a little protein that sort of tethers to the wall. So when you blow by, the enzyme does its job, but it doesn't get washed away because it's tied in with a tether to the wall of the small intestine. Kind of an interesting little feature, right? Again, we're not going to go through and list all of those. There's a lot of disaccharadases that are important here, and a lot of other things as well that are going to help us to digest our meals. But we have a whole bunch of those little guys. It's going to be tethered in here. Just going to tether in here like this. We've got our little tethers kind of hanging, letting them hang on here. And then again, as the digesta, you know, the movement through the gut kind some point, anatomists must have looked at the villi that make up the wall of the small intestine, and it must have reminded them of the bristles on a brush. So we often call this part of the intestinal wall, we often call it the brush border, okay? Because it must have at some point reminded anatomists of the bristles on a toothbrush, right? That toothbrush handle right here. The toothbrush handle, right? Brush your teeth. Like that. So it must have reminded anatomists of the brush at some point. So they call it the brush border. It's a really common thing you can see. And these enzymes are called brush border So the name of the game up here in the bottom is finishing things. One of the things that increase in delivery here, starting to protect, neutralize, and then it's that digestive kind of flows into the jejunum. Those enzymes are doing their job, and they're breaking down the materials into the individual building blocks, they're running into the brush border enzymes through here, we're breaking things down, and then we get to the ileum right here. And now we see that we have a change again. And now what we're doing is we're starting to absorb those building blocks, those monosaccharides and those amino acids and those fatty acids. We're starting to absorb those into the bloodstream throughout the remainder of the ileum, right? So we're going from a place of secretion over here, right? Secretion, 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 digestion, 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 absorption, 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 absorption. Does that make sense? All right. So I get my steps in today too, making it long, small intestine. All right. So what we need to do is I would like to take just a minute, and I'd like us to look at the last part of the small intestine, since we've been taking our tour of the small intestine today. We just looked at the duodenum. We saw it's just chunky jam full of these duodenal glands that we have in here, or these submucosal glands. We looked at the jejunum to start with, and we saw that that's pretty boring. Nice, pretty villi and goblet cells and stuff like that, but not a whole lot else. Now, what I would like to do is I would like to look at the ileum under the microscope as well, right? Now, in the ileum, we're absorbing things into our bloodstream. And so this is where moving things from the lumen of the gut into the external or into the internal environment is where we can potentially run into problems, right? So we're going to see that the amount of safeguards against that starts to increase, right? So we're going to see the presence of these. We're going to see the presence of these Peyer's patches or lymphatic nodules. Right? We're going to see the presence of these Peyer's patches or the lymphatic nodules starting to become really, really prevalent. Keep in mind that these Peyer's patches start in the submucosa and then they sort of erupt into the mucosa. So we're going to see them really kind of taking over the small intestine in a lot of ways. For this, you guys are a small class, which is really good, because I only have seven. Hey, I have seven. I have 7 slides. I have one for every group. That's all I have though. So I have 7 slides, and only in the Pyres patch. They're not in a tray. They're just down here on the bench. So come grab one of those slides. Let's take a look at Ilium underneath the microscope. And let's take a look at Pyres patches.
Saturday and Monday as well.
All right. Just so happen to have 7 of them. Almost like we planned. It's good. All right. So again, we're not interested in every layer here. If I ask you about the layers, I'll pick that very first slide that we started with today. That's like a textbook small intestine. Right? Here, I'm only interested in part of the story on the slide. Right, so our small intestine is kind of going along and going along and going along. And then kablooey, look at all of these Pyres patches that we see in here, right? Again, they literally just kind of take over everything, right? So the Pyres patch starts right here, but you can see that what it's doing is it's just erupting into the mucosa. You can't even really tell where it starts and ends, right? But that's what we're kind of looking at. We've got one right after the other. These Pyres patches will continue into the large intestine. The lymphatic nodules underneath the large liver. These will continue into the large intestine. You have a little bit of absorption occurring into the large intestine. Lots and lots and lots of Pyres patches. we doing? Not too much to see here. Like I said, we're not going to focus on every layer or definitely.